Good afternoon, everyone. Welcome to our edition of Research Edge. We have been running this session for this whole fall semester. Today, we have another two more interesting presentation, two diverse presentation. One is very much focused on the sciences, and they have the second one focused on the social sciences. So I hope all of you all are well and have taken your lunch as we move to our session today. So today we have two presentations. The first one, the potential of using a scanning electron microscopy to advance research in Bahamas. So this presentation is co-authored by three authors here, Dr. Jelena Andrejić, Dr. Paul De Luca, and Dr. Vladimir Pavlović from the University of Belgrade in Serbia. So I understand that Dr. Andrejić is going to do the presentation, but Dr. Paul De Luca is around as well for any, if there's any discussion later. And then you have the second presentation, the impact of COVID-19 control measures on stress in the Bahamas. This will be presented by Dr. Stephanie Hutchinson and also co-authored with Dr. Naomi Hall Campbell, Mr. William Fielding, and Dr. Vijay Perumal. I understand this is a research that is still in progress. So you may not get a full presentation of this research, but still what is important in this research edge session is the discussion so that this study can actually be completed. So without further ado, I am now going to invite Dr. Andrejić to actually do her presentation. I will stop sharing my screen so that you are able to actually share your presentation. So over to you, Dr. Andrejić.
Hello, can everybody hear me well?
Yes, we can. We can see your
Thank you.
screen.
Thank you for the introduction, Dr. Nair. I hope everybody is doing well. So I started this with Dr. Paul De Luca because I'm more of a plant person and I study more of the micro world, and he's more of an animal person, and he, of course, in our department, he is known as a professor who studies entomology, so he's more into insects and animals. So any questions, please direct them to them, and I will answer all the other questions. But this is why I partnered with him, because I was trying to kind of bring this micro world into a micro world, what I study. And also I share, the love of electron microscopes, and especially scanning electron microscope. So when I went back to my home country, I contacted one of my friends. He is a physicist who works in the Faculty of Agriculture, and they just got brand new scanning electron microscopes and a new transmission electron microscope, which was literally in the box and unopened. And so I brought in some samples, and I was thinking of how we can make this into something useful. So before we go into the, like you said, Dr. Nair, the science stuff, just a brief introduction that electron microscopes are very different from light microscopy. These are much bigger, high power microscopes that use electron beams instead of a light. So they can magnify specimens a lot more than just the conventional light microscopes, because we use energized electrons, and we speed them up in the microscope, and that's how you get the image. In addition, this is unfortunately something that we didn't use, but we plan. There is an X-ray analysis that you can do. So, apart from, this high power topographic image that you get, you can also study what is the chemical composition of the sample. Like I said, we were not doing this part as well. That is maybe something we're going to do next, because we were just kind of trying to look what kind of samples are we going to get in our specimens. So it works. I mean, I will try to present it very simple, not taking too much time, but pretty much you have an electron gun, which passes through an anode to kind of bring the electrons and focus them through the lenses. These are not like lenses you will see in light microscopy, which are made of glass. These are like electromagnets to kind of focus the beam on here. And then the secondary electrons, the ones that are bounced off the specimen, are collected here, and that's what we see. And then there is also a second type of electrons which are collected by the X-ray detector, and that's going to tell us, you know, what the specimen is made of. And, you know, the resolution in like new high power scanning electron microscopes can go into the nanometers. So viruses can be easily visible. So in biological research, light microscopy might be used for like some general studies, but a highly desirable tool in modern biology are those high power TEM, transmission and SEM, scanning electron microscopy, for a variety of research. You know, you can study the topography of the cell. You can even visualize macromolecules. So anything that has to do with cell biology and anatomy. Then especially in taxonomy, we know we use a lot of genetic studies to do the structure and to kind of differentiate bacteria, but we see that, you know, we use that in taxonomy as well, especially in bacteria, fungi, algae, plants, insects, and many other organisms. And we can also use scanning electron microscopy in anatomy studies to study, you know, anything that's like calcified, like teeth and bones. So, and I mean, the list will, you know, just go on. Really the usage of this microscope is pretty wide. So this is how the lab was set up in the Faculty of Agriculture in Belgrade, which is the major capital of Serbia. So this on the left-hand side would be the microscope. So you see the electron gun is quite huge. There is also beneath here a large reservoir of liquid nitrogen to keep everything cool. With the biological specimens, you know, because you're shining, and I mean, you're bombarding the soft tissue with electrons. If you don't protect that with some type of coating, you know, the whole specimen will just start to melt. So what is done with the biological samples is that they need to be coated with gold. And I remember I was sitting here with my coworker, and we were joking about, you know, like these machines are not expensive, but it's the gold that is needed for this so-called gold sputter to work. So we were joking, you know, is anybody willing to sacrifice a wedding ring for the research? The electron microscope, that's an expensive part, but like I said, for biological research, you would also need this. So what we have done with our samples is you have to mount them on this stub, and then you kind of gently glue them on, and you put them in the gold sputter, and you see everything is sputtered with gold. So you know, you get nice gold shiny insects that you then put into a chamber, and everything, of course, inside the chamber is under the vacuum. So you put the microscope, you put the specimens inside the microscope, and now this thing can rotate, not a lot, but it can kind of, you know, rotate to have the better angle of visualization. We had several specimens of insects, so we will only be showing a couple of those here. I also brought some algae. Again, we will be looking at this one, and I collected some sand samples from the Eleuthera island, so that's what we will be looking at. Those are going to be microphytobenthos that live in sand. And we also have a flamingo feather right here, as you can see, that we put underneath the microscope. So we have a lot of images, you know, like when you sit down on the microscope, I mean, literally the whole day can pass, and then you end up with like hundreds of images. So we kind of pull out the really interesting ones. So what you can see here in this image, this is the stinger of a wasp, and I believe it's called a paper wasp. So this is a modified ovipositor, which is used to lay eggs, and as you can see, it is also modified to be like a weapon. So it literally, it's like two spears that form a hollow tube inside here. But then, you know, when you magnify that even further, you know, you look at something very scary, which are these forward-facing barbs, you know, to make sure, you know, when you get stung by a wasp, that, you know, the venom can go, and I guess that this ovipositor doesn't kind of slip out. So it can have a firm grip on your in your tissue and your skin. So ouch. So that's you know, something that is visible when you use like a standardized microscope, and I'm sure Dr. Paul De Lucas shows this to the students. But you know, once you see this underneath the 3-dimension SEM micrograph, you know, you really kind of get the appreciation of how fascinating this structure is. Another thing that we looked at was a modified rear leg from a common bee. And this structure is used to collect pollen. It's called corbicula. We can even see, you know, these are structures, pollen presses and rakes, which are used to compress pollen so the bee can carry as much as pollen as possible back to the hive. And in addition to that, when you magnify even further, you can see, you know, the branching bract hairs, you know, that help track and hold even more pollen. So it's really fascinating, you know, when you kind of go from low magnification to higher, and then seeing this in very, very fine detail. Another interesting structure in insects, we were looking at this a longhorn beetle, and this is a part of his head. So these are the compound eyes. So the eyes of an insect are made out of many smaller eyes, and these are called ommatidiums. So that's how insects see. Pretty much each eye function on its own, and then collectively they give an image. And what I really like about the scanning electron microscope is you know, you can really see the 3D structure, and then there is also a scale bar, and this is really, really important because you can use another computer programs to kind of maybe measure individual ommatidiums, to maybe kind of study the structure. Is there a difference between the left and the right eye? Is there a difference in size between females and males, or maybe closely related species? So you can really use these images for doing pretty good research. With the algae, this algae is most likely Turbinaria turbinata. So this is the close-up of the its thallus. Now, what we see here are probably a mixture of mucus glands. So these are probably, you know, channels through which some extra mucus is secreted. And some of those are called conceptacles, which are fertile cryptostomata. These were once reproductive cavities. Now, these structures are not fully understood because the way this algae reproduce is it produces modified leaves called receptacles. So, you know, these are kind of the leftovers of how some of its closest relatives used to reproduce. So there is a great research to kind of try to find out what really those structures are. So here you can see, you know, their detail, and here is the scale bar. So this is like 100 microns. So this is a close-up of the conceptacles. Another thing that I've managed to collect is the calcified talus of red algae, of coralline algae. This can be used in studies. I've been doing a lot of research, right here you can see is that this is where the limestone is deposited by a single cell in red algae, and that's how they build their skeleton. So very, very similar to corals, although honestly, when you look at corals underneath, I mean, when you look the skeleton of corals underneath the scanning electron microscope, it looks even better. But it's the same buildup that you can see here. And it's very similar to a honeycomb, but this is where one epithelial cell of red algae will kind of grow and deposit the hard crust. So why is this important to be looking and measuring underneath the scanning electron microscope? Now, these two images are not mine, but I'm sure that you all heard of bleaching of the corals. Also, algae can bleach, and they can bleach due to changes in the pH of the water. Especially, they're very sensitive to changes in the temperature and the acidity. So what is happening is that the limestone doesn't get deposited the correct way. So you will see, you know, that there are like large holes and grooves, and where like this space where only one epithelial cell should be, you can see like it didn't, like the dividing wall didn't form properly. So you can kind of you know, study the effects of warming and worsening of the water quality by looking and measuring these structures underneath the scanning electron microscope. So now this really opens doors for many types of research to be done on different types of red algae. We can study different islands. I mean, like really the possibilities are endless what we can do with this. So just, these are not my images. So this is from another paper, but looking at what I've collected, it looks pretty healthy. I don't see any major, you know, this is a young algae, so you see the grooves are not deep. So, but I don't see any anomalies. So I guess the waters around Eleuthera are pretty clear. Then in the sand, we see a lot of single cell organisms, and foraminifera are kind of closely related to animals. They build their shell out of different particles. Many of them, they build their shell or house out of calcium carbonate, and this is how they look like. They have a very great diversity. They're also used a lot in paleolimnological studies. So, you know, if you were to dig and excavate a core, you can see the foraminifera, and then based on the species that are found, we can reconstruct what type of environment was 50, 100, up to 500 years ago. And the only way that these can be identified is you would need to have a scanning electron microscope, because underneath the conventional microscope, so with the light microscopy, again, this is not my image, but here you can see how they look underneath normal light microscopy. So really you cannot identify them like this. You would need a scanning electron microscope. Then we have diatoms, which are single cell algae. They are very abundant, found everywhere, very common in phytoplankton. What we see in sand are those that like to burrow through sand. And we also see those that are part of the phytoplankton community but have died and sinking down to the bottom. And they're fascinated because of their shell, which is made out of silica, and it's called a frustule. And then enable for them to exchange the nutrients with the outside environment, you see these pores called puncti. And also there is a slit which enables them to move through the same grains. And these puncti are really, really small. They, range up to nanometers. And there is a great interest in nanotechnology for these algae because, we don't know the applications. There is no laser, I guess, that they can cut so many holes within the nanometers range. So there is a great interest for these in nanotechnology, and their structure can vary from like being very simple round with bigger holes to some very complex, like we see in this Surirella that has wraith along these bridges which are suspended. So some of these diatoms have very complex structure. We see a lot of sponge spicules. They're literally everywhere. Some of them are less branching, some of them are more branching, and some of them are quite small, as you can see here, but they're literally everywhere. And then we have these, which we don't know what they are, unfortunately. Maybe this can be a cyst of a foraminifera, but like at this point, it's like sometimes you find some things that you are not sure what they are. This is an interesting image where you can see, one of the spines broken off, and there is a hollow tube leading into the center. So I was not able to identify what these two are. And then you have the flamingo feather. So this is a mature flamingo feather where you can see a center barb with two types of smaller ones called the barbules. And then you have the anterior and then the posterior. What you can see in here is that the anterior barbules possess structures to help intertwine and interlock with the neighboring barb. So this is very important for for these birds to fly, for the feathers to be interlocked together. So the general idea that we had was really to make an illustrative atlas. So we would have images which would be colored. Now, all of the images that you've seen here, they are in black and white. So the images that are produced with the SEM are black and white, but these can easily be, colored, and to kind of get, more in-depth perspective of what kind of different structures are in there. So our general idea was to get more samples, maybe a little bit more of the plant material, such as pollen, maybe some fish scales, maybe some Bahamian sharks scale. So whatever we can find that we would put underneath the electron microscope. And really this is a way to kind of popularize Bahamian biodiversity in a very different way. So with this atlas, when you look at endemic fish, to some people, I guess it's just the fish. It doesn't really mean nothing. But when they go through this atlas and when you go through these images, they might see the biodiversity from another angle, another perspective. So that was one objective we would have in mind. And then another thing is, once you have a collection of these images, you can use that for different types of morphological study research, to highlight or to describe if there are any new species. You can use it in anatomical studies and so forth. So there are a lot of possibilities that you can use. So a lot of collaborators that can be summoned into within this project. Unfortunately, we know that the research fund was suspended for this year, so we are waiting. We're going to gain. I'm going to get more samples and hopefully have more images, so that we can have this illustrative atlas hopefully printed out. I would really like to thank two people. It's Paul De Luca first for providing me the insect samples. So thank you, Paul, so much for that. And I would also like to thank people who helped me with looking at these images underneath the scanning microscope, and that's my colleague, Dr. Vladimir Pavlović. So I guess that is it for me. Sorry if I passed the time. I thought I was speaking very fast. So that is that for me. I'm just trying to stop share. Okay, there we go.
Thank you so much, Dr.
No
Andrejić.
problem.
I think it was really a fascinating presentation, fascinating images. So there are a few questions in the chat. I think the key question that I see is, what is the cost of an SEM, and is it something that a university like the University of the Bahamas can actually afford?
It's expensive. I mean, as an alternative, maybe buy like a used one. But from my experience, because I worked with new one, which was at the Faculty of Agriculture, and then I use a very old one, used one, and it's a lot more maintenance if you purchase, you know, used ones. So it's really expensive because especially for biological research, you would need the gold sputter, and then you need gold, and then you need to have liquid nitrogen. So it's I mean, yes, in the future, but I think it's always best to kind of have samples and have collaborators, and just you kind of literally sit down one day and you take pictures endlessly. This is the scanning electron microscope is really maybe for more bigger institutions because it will be used way more. When I mean, this is like the Faculty of Agriculture, and this microscope serves, I think, like 5 faculties, 5 research faculties, and I just sit there whole day. Literally, it was all day for me.
Are you aware if such microscope is available anywhere in the Bahamas?
No, I haven't heard of any. No.
Okay.
Or TEM. No.
Dr. Amin Kabir, you have a question?
Yeah. Thank you, Vic. And yeah, it was a very good presentation. I really enjoyed it. So I have one question. The pictures that you have shown from the Bahamian sample, those are all topographical, right? Like using secondary beam. Is that right?
Yes,
okay.
That's right.
And you said what was the resolution of the pictures?
Well, like I said, we didn't. I think this was the highest that we. No, maybe it's not. What? Let me see. What is this? Is 100. Well, if I go through, but I remember that I even magnified this even more, and I went to 1 micron. I don't remember going any further than that because we didn't see anything that was interesting to us even further than this.
Okay,
right. So this is
but what was the okay? So what was the resolution of the SCM that I mean your collaborators bought? I mean, is it like several nanometers, kind of in the range of nanometer?
Yeah, definitely in the range of nanometers. Yes.
Okay, yeah, go ahead.
Okay. I was about to say that this one that they bought is relatively new one.
Okay.
So it can go nanometers. Yes.
And you said that you haven't yet analyzed the X-ray diffraction one, right? Because if you do so, probably you would be able to know the chemical composition. Is that right?
Yes, that is right. Now, we didn't use that because this was all on my expense. And I actually had to, like, if I was to do that, I would have to buy the liquid nitrogen, which was like expensive. So that's why I said, okay, so I kind of skipped that part. Okay. But for next time, if I had like funds, then basically that's the only thing that you pay. Like I didn't pay Dr. Pavlović is my close friend, so he
Yeah, so you
didn't
get
pay.
the picture, okay, of the surface picture, basically didn't get.
Right. But
Okay.
so the only thing that we would have to pay was for the liquid
Right.
nitrogen to
Nitrogen,
cool
yeah.
the X-ray gun. Yeah, yeah. So we didn't
Yeah.
do that. But that, can you now imagine how this can open, a lot of possibilities for different types of research now that you
I can
can
see.
see an image and plus know the chemical composition. I mean, it's pretty amazing.
Yeah, so far I understand that how you're gonna really utilize the Bahamian source and take it to probably your collaborator and get the images to know that the formation, like when those are formed, like by just looking at the picture, like if there is any acid effect or how long ago those are formed, that could be done, right? I mean, other than that, what else you can do from the Bahamian sample?
I would really like to look at some coral samples. Because again, those groove can tell you whether the coral is laying down the calcium the right way. I guess that would be something that I would like to look at, collecting some coral samples. And I would also like to look at pollen samples of an endemic plants. And I don't know, Paul, is there anything else that might be on your wish list that you would want to see? I'm sure that there is plenty of stuff. Like literally the sky is your limit, what we can do. This is for the biological samples. We can also do some geographical studies of the soil, because usually it doesn't require coating with gold. So you would just, shine a beam of electrons. So this is just for biological specimens. So it might be a little bit less expensive.
Yeah, you said that you gold spatter for all the samples, right? All the biological samples. And
yes.
Was there any problem, like, because I think you gold spatter because to amplify the signal, and also probably the charge accumulation problem kind of thing? Is that right?
There was a problem with the bigger insects. They would have to be dry.
Exactly.
Because if they are just a little bit wet, then the beam will pass through the gold coating and pretty much, like the specimen will start to melt in front of your eyes. Like you can see that on live TV, it's melting. So everything would have to be very, very dry.
And it also, because you are putting it in a high vacuum chamber, right? Because inside, I think it's a high vacuum that you use, right?
Right. Yes. Yes. Yeah.
Okay.
It takes like at least 10 minutes to vacuum that thing.
All right. Thanks very much. I had some more questions, but I can always discuss with you.
Yeah, thank you, Dr. Kabir. I think there's another important question that I see here on the permit that is required when you are when you're exporting sample to the lab in Belgrade. How did you get the permit?
That's
what is the process? Because this is something that is critical for us to understand, because there's a lot of challenges in getting the samples out of the country.
Yeah.
Right. So when I exported these samples, then the law was passed literally, I think, like a week after. So I was already gone. And then the thing is that all the samples were actually dead, like nothing was alive. The algae, the insects, these are dead specimens.
Okay.
So they're not, it's just like calcified algae. And so nothing is like alive. However, we would, I guess we would have to contact the ministry. Like if we need to like have a permission to export a tiny piece of coral, like I don't know, but
I think there's a new act now on controlling this, and there's not BEST Commission, but now it's under the DEPP, which is the Department of Environmental Protection and Planning. And in fact, I think next week there's a meeting that they're calling UB to discuss on this, because I think there's more control now on researchers coming in to places like Jerez and carrying out research and taking samples out. So this is there's going to be more control after this.
Right. The only thing I hope is that like we are not transporting anything that's alive. So everything would be dead. So
All right. So thank you so much.
thank you.
It was really a great presentation, and we really hope this research will further expand for us to get these fascinating images and publish all these images into our own the Bahamas database, and we hope this work will go through, and we hope to help you once the fund gets moving and we get more funding to get this project moving. Thank you so much.
Thank you.
All right then, so now we move on to our next presentation, the impact of COVID-19 control measures on stress in the Bahamas. So we have both Dr. Stephanie Hutchison and Dr. Nyambi Hall-Campbell who will take us through in this presentation. So you're free to share your screen for your presentation. Over to you.
Okay, that's the ice cream truck cruising through my neighborhood. Good afternoon, everyone. Very happy to be here. My first presentation for Research Edge. Thankful for my colleagues who joined me, Dr. Hall-Campbell-Dean, Mr. Fielding, and Dr. Perumal. Thank you so much, and thank you for this opportunity. I'm just going to put the slide here just for a moment because it's in more detail in the flyer. But so just to get started, and then move into the introduction for this presentation. Lockdowns, quarantines, and restrictions. Oh my! This is our 2020 reality. We live this. We lament about it, we read about it, and in some way we've all been impacted by these control measures to reduce the transmission and the impact of the coronavirus. And as a psychologist, I'm particularly attuned to the mental health impact. So take a look at these global and regional headlines that speak to it as well. WHO, facing mental health fallout from the coronavirus. That was in May. Prediction fairly early in the global reach of the pandemic. And then a growing number of people facing mental health fallout. That is out of Santa Barbara, California newspaper. And then more closer to home from Paho, the Caribbean region specifically, facing mental health crisis. It's kind of like an entree into this presentation. And so our objectives was just to assess the change, stress in residents of the Bahamas from the pandemic. As I showed you these slides, I'm also reminded of the inspiration because I've been seeing so many headlines about the mental health impact, the mental health fallout, and I didn't hear too much conversation about that here, except among my colleagues. And so our interest was, well, what's happening in the Bahamas? How are people responding, particularly in terms of stress, to the continuing saga of the pandemic? And so just to I guess, get us on the same page with some key terms. They're not on the slide, but I'll just speak to them. Some of the key terms for this presentation will be mental health and will be stress. My new mantra is mental health is health. Mental health, by definition, is our emotional, psychological, and social well-being. It affects how we think, we feel, and we act. Mental health helps us in terms of how we manage and handle stress, relate to others, and make choices. And that's a very simple definition from mentalhealth.gov. Stress, in terms of another anchoring term, stress refers, and this is the easiest term that I found, easiest term for the general public. Stress refers to how we adjust to changes in our lives, whether they're positive, good changes, or negative changes. Those are key terms to anchor us. I'm sorry that part of the slide did not show up here in the translation to my iPad. But what I'm speaking to here is the significance of the study in the need to place emphasis on mental health and COVID-19, the recognition of the negative impact of mental health, and prioritizing services to include in policy decisions. And so just to expand upon this piece here, health agencies and multinational bodies, prominent peer-reviewed publications like The Lancet and the British Medical Journal, professional organizations like the American Psychological and Psychiatric Associations have been quite vocal in the need to place emphasis on mental health in the COVID-19 pandemic. As an example, in May 2020, the Secretary General of the United Nations, Mr. Antonio Guterres, spoke about the negative impact of COVID-19 on mental health. And this is what he said. The COVID-19 virus is not only attacking our physical health, it is also increasing psychological suffering. Grief at the loss of loved ones, shock at the loss of jobs, isolation and restrictions on movement, difficulty in family dynamics, uncertainty and fear for the future, end quote. The World Health Organization, WHO, concurs, and they've also raised concerns about mental health. These organizations agree that mental health matters should be prioritized as an essential part of all government response to COVID-19. So while most emphases have been placed on interventions to reduce the impact of COVID-19 transmission and infections, provide medical care, procure equipment needed by healthcare professionals to safely perform their duties, meet survival needs, all of these are critical. The mental health needs tend to get the short shrift. Therefore, it is vital, underscoring the significance, it is vital to incorporate mental health, and particularly for this research, stress responses, as a critical metric for health. When we recognize this, then it allows for more resources to be deployed in this area. And it also means getting creative with the what and how we meet this very real need. And there's data to support what some in the mental health community are calling the silent and parallel pandemic because of its global impact. That is the mental health fallout. Rapid review of the literature and published research provide evidence for high incidences of depression, anxiety, and suicides associated with COVID-19. Additionally, those with pre-existing mental health conditions have also been impacted by disrupted access to needed services and support. So it appears that no segment of the population that is immune to negative sequelae associated with the restrictions, the limitations, the uncertainties, and the disruptions that have characterized the global experience and our experience of COVID-19. For certain, we've all made adjustments along the way, but with the loosening, then tightening of restrictions, and the constant flux and disruption to our lives since March, when the first case was identified in the Bahamas, it has been a tumultuous journey. It's been wearying. It's been stressful. My colleagues and I mentioned earlier, Dr. Hall Campbell-Dean, Mr. William Fielding, and Dr. Puramal, conducted research examining the stress impact of COVID. We completed the first wave study in May, and then completed data collection on the second wave impact in September. We'll speak primarily to the first wave data. For the first wave, we looked at the 30-day period, inclusive of the Easter 2020 5-day extended lockdown and ending May 29, 2020. So that's when we cut off data collection. We used two standardized scales to assess stress: the distress questionnaire, DQ5, and the impact of events scale revised. The DQ5 is a 5-item measure commonly used to assess psychological distress and has efficacy for community-based research. The IESR is a 22-item measure often used to gauge stress responses to traumatic events. The DQ5 was completed by all respondents, while those who identified as frontline workers also completed the impact of event scale revised. Using social media, snowball sampling was used to solicit participants in the study. Preliminary findings from over 700 participants on psychological distress have been instructive, and I will turn it over now at this point to Dr. Hall Campbell-Dean, who will share the results and implications.
Thank you, Dr. Hutchinson, and good afternoon, everyone. Okay, so when we look at the results of our study, we want to just start with our demographics. And so we see that overall, we had 702 respondents, which we were really excited about. Overall, 24% of the respondents were male. So our respondents were overwhelmingly female, which is not uncommon in regards to results, especially in the manner that we engaged our methodology. The modal age group was 45 to 54 years of age. And in addition to the 62% of our respondents that were employed before COVID-19 crisis, an additional 12.9% reported that they have been negatively affected financially as a result of the crisis. So we just wanted to highlight that because employment and finances are big factors when it comes to stress levels. And we also wanted to highlight our age distribution because the stress levels were connected with, or we did see differences in regards to age and the stress levels. Next slide, please. Oh, sorry for the formatting. But this is our third table, and this showed that the level of stress of all participants prior to and within COVID-19 for the DQ5. And so what we can see from this is that the overall percentage of high stress respondents increased from 16% to 25% from the pre to the during COVID-19 period. So as you can imagine, people were reported being more stressed during COVID-19 than pre-COVID-19. And so you may say, well, you know, that seems self-explanatory or obvious. But one of the reasons why we wanted to conduct this research is so that we can actually have data to support or implement programs in regards to mental health, right? We don't want to just say, of course, people are stressed out. We want to be making decisions based off of the data. So we have data to prove that stress did increase during this COVID-19 period, okay? And so one of the interesting findings that we see in our results, we talked about the ages. And so you see the level of stress for all participants prior to and within COVID-19 lockdowns by age group. And so we found significant increases in stress with the 45 to 54 age group, and then the 55 to 64 age group. And so this was what we had expected, specifically with the 55 to 64 age group, because if you remember during the early lockdowns, a lot of emphasis was placed on people who were vulnerable to catching COVID. And the older populations reported, or the older populations were cited as being the most vulnerable. So that's why it's kind of interesting that our 65 and plus age group didn't have a statistically significant difference, but we also know that their numbers were pretty low in the overall sample that we had. So that might be what is happening there. You will also want to note that the change, oh, well, we could see it here, but You can go to the next slide again, Stephanie. The change with the young people, although it was not statistically significant, young people ages 18 to 24 years old experienced the highest levels of COVID-19 stress in comparison with the other age groups. And so we thought that was really interesting because this was the age group that was supposed to be the most resistant, right? The strongest. And I don't know if, it seems like we've been talking about COVID for years now, right? But in the beginning, there was even kind of this notion that young people couldn't get it. But in our sample, we found that they experienced the highest levels of COVID-19. And so we thought that this was really significant because a lot of the measures and suggestions that were being made were speaking to the fact that you don't want to catch this, you don't want to catch this. But these results show that even though these young people were probably resistant or most resistant to being subjected to COVID-19, that they were still experiencing high levels of stress. So that the stress that they maybe have been experiencing was separate from the stress of actually transmitting COVID-19, right? We found studies that talked about the stress of college students doing online classes, the stress of trying to find employment in an already tight economy, right? And now they see their parents either being temporarily laid off, furloughed, or having reduced hours. And so their options in regards to, you know, what they could do for employment probably seemed even more dire. And so one of the significant discovery along with that specific highlight is that the majority of those surveys demonstrated higher levels of stress during COVID-19 than pre-COVID-19. And we wanted to, you can't really see our take-home message.
I could, yeah, the white print. The take-home message: policy and societal responses to COVID-19 must mitigate the deleterious effects of health on overall health and well-being. So that was the take-home message. And our final slide.
And so I see we're winding up on time, so we'll just end there if there are any questions.
Yes.
Thank you so much for the presentation. I shall open the floor for any question. I don't see any question in the chat. Is there any question from the participants? How far are you in your analysis of your result for the second wave?
Well, we've done enough analyses to note that we've doubled our participants. We went from 700, a little over 700 to 1,400 respondents. So that was really good. And we did that within almost like a 48-hour period. We had that rapid response. So what we know from the second wave is that people are still reporting being very stressed, up to, I think, 49%. And they also are self-reporting that they are more stressed in the second wave than in the first wave. And also we have almost 50% of them who are also more worried about catching COVID-19 in the second wave than in the first wave. So as the cases have ramped up, so has people's level of stress and concern about catching COVID as well.
There's a question in the chat. Is a lack of social interaction causing the reduction in mental health?
There's a lot of research that is supporting that, because social support has always been considered a protective factor, whether it's mental health or physical health or just for general well-being. But the very strategy and tool we use for support is also the tool that can cause us to be exposed to COVID. And so, whereas before, when someone is ill or there's some challenge, you come together in community support, but because that's connecting with people outside your household, the social distancing, it's made us further apart. And so the traditional measures to fall back on to help support and strengthen our mental health kind of has been pulled away. And so we have to find different ways, trying to stay connected virtually to kind of fill the gap there.
Okay. Do you think that the increasing stress levels can worsen the illness of individuals that have COVID?
Well, I think that was one of the main reasons why we wanted to conduct the study, right? As Dr. Hutchinson stated in the beginning, mental health is health. Yeah. And one of the things that we know about stress is that it depletes our immune system. Absolutely. And so if your immune system is weak, we know that that makes you more susceptible to catching COVID. And so if you already have it and your immune system is weak, that makes you more at risk for, you know, your symptoms of the disease being more severe. And so this is why we are advocating for the recognition of mental health being purported as we talk about COVID-19 measures, right? Because it's not just about not catching the disease, but it's about how we can do things to prevent illness in general.
Yep, indeed.
Right. And I think many more of us, I mean, think about us here today. I was sharing with my colleagues as we were building up to this. As we're in week 13, this has been the most stressful first semester for me. It's been stressful because the layer of COVID has contributed to that, trying to kind of pivot and teach online and kind of juggle all of these balls and just trying to find my rhythm for how the semester would work added a level of stress because I'm trying to function in COVID. Our students are reporting some of the challenges that they're having. And I think we all have, at various levels, we're adjusting and coping with stress because it's wearying. This has now become a chronic stressor. It's no longer acute, it's chronic. And so we have to kind of think about what the long-term impact is going to be. If you look at, as we were looking at the research related to the SARS epidemic, the research from the SARS epidemic speak to the fact that even years later, once the transmission and the virus was contained, people were still feeling the mental health impact. And so we have to pay attention to that body of research, because I think it's instructive to let us know what we could be potentially looking at even once the vaccine comes on stream.
Yep. Okay, any more questions? All right then, so if there's no more question, thank you so much, Dr. Hutchinson and also Dr. IMB Hall Campbell for the presentation today. Indeed, we have two diverse presentations and we've learned quite a bit today. And we hope you will actually complete this study. I think this is an important study that has to be published. And we hope, in fact, both the studies that we've listened today, there's a lot of potential for it to be moving to the next stage. And we really do hope we are able to support as much as possible for you to complete these studies. So with that, we will conclude our session today. Just a reminder, our next Research Edge session is going to be on 4th of December. So it is quite soon. This is going to be, this session on the 4th December is to celebrate our 20th anniversary of the inception of Research Edge. So the Research Edge has been around 20 years. And we're going to have a short presentation on analyzing the trends of research that was presented at Research Edge. And it's going to be an interesting session. You'll see the flyer coming out on Monday. So I do hope you register and come for this session. The session will be officiated by the President and also the VP of Academic Affairs. We will also have a toast at the end of the session to celebrate the 20th anniversary. So please do register and come for this session when you see the notice coming out soon. Okay, let me see. Before I end this, there's some messages coming in. All right. Okay, there are still questions for the second presentation. During your research, did you come to a conclusion on ways in which a person can decrease stress levels, like ideas which can help individuals adapt to the new norm?
Because of the nature of the measures that we used, they were very short and they were more, they didn't really ask questions specific to that. And so that's where the third and the fourth study is going to kind of look at specifically why are people stressed the way they are, and what factors have been protective for them and what worked for them in terms of helping them manage stress. We know from the research social support is a good one. Routine, I consistently see in the research that maintaining a routine and having a routine has been one of the strongest factors that help people to have some sense of stability and predictability in being able to cope with the constant flux associated with COVID. And so having a routine, making sure there's downtime, protecting your boundaries, maintaining support, even if it's virtual, those are some of the ways that people can fare better in this COVID environment.
Thank you so much. So with that, we would conclude our session today. So have a good weekend, stay safe as always, and hope to see you all again in our next session. Bye-bye for now.